And I think that we are in a very special session, and urban lakes can be not a target study for most limnologists, but for many of us, they are in the way of our life, near institution, near our residences, and they are very important, besides not so much studied. For instance, here is a lake inside the botanical garden of Rio de Janeiro city, which is inside the Rio de Janeiro city. It's a very beautiful place, and most of the visitors would like all the time to see this lake because of the aquatic plants. Inside this pond, there are Victoria regia. It's a very beautiful aquatic plants. So this pond is very, very important for the scenery of the botanical garden. And I think that is one of the example of urban lake that we should have be studied mostly. Well, shallow urban lakes are important part of the scenery of many cities across the globe. And differently from other lakes, they usually have more people coming in contact with them. For instance, this lake is so attractive that we have many guards looking after him because people would like to be in contact with water, and this lake is prohibited because it's a very protected area of the botanical garden. Urban lakes can be deeply associated with local landscape and are frequently important for aesthetic improvement, as well for preserving local and even regional biodiversity. The frequent existence of eutrophic and hypertrophic conditions in the system has been a challenge for researchers and local municipalities. Everywhere, the challenge has been to apply efficient strategies for management and recovery of these shallow lakes. This is another shallow lakes in Rio de Janeiro city, near residence areas and very, very high eutrophicated. Well, one of the major tasks relating urban lakes has been to integrate surrounding resident people for sustainable conservation activities around these ecosystems, involving water quality improvement and conservation of native biota. And I think that the suggestion for this session was to encourage discussions about these ubiquitous and most often endangered shallow urban lakes. So the schedule of our session will follow the schedule in the spreadsheet on the website of the shallow lake. We will have 9 presentations. So we have about 10 minutes or more for each presentation. And we can see from the titles of the presentation that we will focus on several important features of this shallow urban lakes, such as indicators of ecological integrity, process of eutrophication, primary production, satellite methodology to follow chlorophyll values, and the study of some specific communities, such as rotifers, dynamics of potentially toxic cyanobacteria, internal phosphorus loading and macrophytes distribution, dominance of phytoplankton versus floating aquatic plants, and rehabilitation of eutrophic subtropical urban sandpit. Our procedure will be we are going to present a summary of the research, the presentations, and the presenter can complement our summary if necessary, or if something we do not include. So, first of all, the study will be presented by Bernadette, Pinel-Alloul, and colleagues. One of characteristics of our session is that we have diversity of subjects, as well diversity of countries. South America, Mexico, and Poland. So we are going to see different and very interesting shallow lake urban. So, indicators of ecological integrity and aquatic biodiversity in urban water bodies. This study focuses on 20 urban water bodies, natural and artificial, lakes and wetlands of the city of Montreal, Canada. The potential of metrics based on species richness and taxonomic composition of phytoplankton, zooplankton, and microinvertebrates community as useful bioindicators to evaluate biodiversity were tested. The study showed that urban water bodies in Montreal support high diversity of plankton and benthic organisms and are essential habitats for sustaining aquatic biodiversity and local and regional scales. To evaluate ecological integrity, the water bodies were ranked based on trophic status indicators, total phosphorus and chlorophyll, and biotic indicators, species richness and compositions of planktonic and benthic communities. Aquatic vegetation in littoral habitats constitutes an important refuge for biodiversity, and temporary ponds offer unique conditions for very rare species. The best bioindicators for future assessment of biodiversity, conservation, and water quality of the urban ponds were selected and recommended as best conservation practices to maintain a vegetative littoral area, various types of ponds, and an optimum management. So we are open to questions.
So if the authors want to say something or complement.
I will complement the summary, okay?
Okay.
Okay. It's a study that began in 2010 and 2011, and it has been a lot of study done on zooplankton, phytoplankton, and macroinvertebrate, with a more detailed approach using functional traits, using phylogenetics, and very sophisticated studies that are already published in urban ecosystem and in biology and conservation and different kind of journals. Now that I present in this conference is like a wrap-up of all the data that I collect on different community. I would like to give a food web perspective and the general indicators that can be used by manager and the people that are working on those water bodies.
Thank you, Bernadette.
Yeah.
So we have questions. I cannot see the chat. Pete?
Yes, okay.
Hello, Beat.
Ça va?
Bonjour. Bonjour.
I have a question. It will be perhaps easier in French, but we'll do it in English, yes. You say that your water bodies are essential habitat at the regional scale. And we did also a similar study in Geneva, but for us it was not really the case because the urban water body were a kind of subset of the regional scale. You know, they didn't have unique species. So my question is here: do you have in your water bodies some species that you find only in Montreal, in the city, or do you have also only species that you find in the regional scale, that mean in water bodies that are outside?
Yes, I have a lot of study done on the natural lakes around Montreal, in the Laurentian area, in the eastern countries, you know, that are not clearly natural because they are also impacted by residential villégiature and so on. But I can say that some of the species I found in the temporary little water bodies in parks, I did not never found them in natural lakes, like Scapholeberis and Cymocephalus, because probably they could exist in natural lakes, but they will be in the vegetated area or in the littoral zone that I did not sample for the big survey, you know. So I could not say that there it could be a nested community in a bigger meta-community landscape approach, you know, because but some of the particular temporary ponds, they are like a priority effect, and they are like a laboratory system for one or two species. That's all, you know. That's very interesting. Yeah.
Yeah. Okay. Thank you.
More questions?
Yes, please. Alfonso Lugo from Mexico. Hello, Bernadette.
Hello, Alfonso.
I want to know if what is the trophic state that prevails in your lakes? Are they mainly eutrophic?
Yes, there is some eutrophic. The lake, the phosphorus level are between less than 15, more than around 200 microgram per liter of total chlorophyll. So there is a kind of oligotrophic, mesotrophic, and eutrophic systems.
Thank you.
I would like to complement the question. The eutrophic system, the input of high content of phosphorus or nitrogen, it's a natural process, or the nutrients are coming from the surrounding area?
I would say that in some cases they are coming from the surrounding area, because it's like runoff of water, you know, on the residential landscape. But in some of the other, like the wetlands, they are more natural, because it's wetlands in recreational part, no lot of residents or people around. So it's a both of them. Okay.
Okay.
The most impact by the highest level of total phosphorus, there are those that are sedimentary. One is in the city of Montreal, and very polluted system.
Okay. I have to say that your research was an inspiration for me.
Yeah,
because,
thank you.
Yes, I think that we should do this in Rio de Janeiro city. We have many ponds and lakes not yet studied. And
yeah, because this study began when I attended a meeting of the city of Montreal, where they present the water quality data they do for the monitoring, but they were speaking about biodiversity. I said it's not biodiversity, it's water quality monitoring. And I could do the biodiversity on the same systems you monitor for the water quality. It would be very interesting. Now I gave all the database to manager on like a fish. You know, fish is like a small summary for each water body. They have all the data on trophic indicators and the biological components. So now they can do something with that. I was expecting to do like exhibit for each, but it was not done because I don't have enough feedback from the city of Montreal, you know.
Oh, and I was another question, not question, but I was wondering, you considered the richness of taxa, and I saw at your figures, at your graphics, that you have many information on the quality of the composition of the communities.
Yeah.
So my question is, should be better to explore, for instance, the composition of the rate among rotifers, cladocerans, and copepods, or the presence of cyanobacteria, in order to try to find some indicator groups or even indicator species? Is it possible?
Yes, it is, but it has been already done in other papers. So yeah, specific papers for the microphytoplankton has been published in Waters. The specific papers on zooplankton is in urban ecosystem. They were published a few years ago, after 2015 until now. It would be my last paper, I think so, on this project.
Okay, thank you.
So I can send you all the specific paper.
Please.
Yeah.
Please.
Okay.
Thank you.
Yeah.
Thank you very much. So, having more questions? So thank you, Bernadette, for your reply for the questions. And the next presentation will be different process of eutrophication in shallow and connected artificial lakes located in an urban area, presented by Heitor Lemos Martins. Is Heitor on board?
Hello.
Hello, Heitor. How are you? So, from the University of the State of Minas Gerais, Brazil. 3 shallow reservoirs connected by a first-order river in the urban area of the municipality of Frutal, Minas Gerais, Brazil. First hypothesis, similarity of the eutrophic condition. Second hypothesis, increase in eutrophication over the study period. 3 years with monthly data. The principal component analysis showed a discrepancy between the first and third years of study. The PCA also showed difference among reservoirs. Progressive eutrophication occurred from first to third year. Greater discrepancy between reservoirs 2 and 3. Reservoir 2, the only one having macrophyte banks, remained less eutrophic, and reservoir 3, which receives an additional fluvial drainage, had the highest eutrophication level. So do you have question for?
Yes, I have question. Do you see me? Okay. Okay. I have question. The study is mainly focused on.
Just a minute. Hater, could you turn on your screen so we can see you maybe and we can interact with you?
Your video.
Your video is okay. Okay.
Oh, thank you.
We see you now.
Nice to meet you.
Thank you for the nice diaporama and the nice presentation you have prepared for the Congress, you know. And I was wondering, you were looking at 3 different kind of reservoirs in the urban area, and you were looking mainly on the water quality, physicochemistry variables, you know. And you said that there is some difference in macrophyte presence absence among the reservoir. And what I would like to that you expand a little bit on the effect of macrophytes. You know, there is only one reservoir with macrophytes, and this is the one that is less eutrophic, you know. And could you explain a little bit about it, you know?
My English isn't very good. I've misunderstood. Happen some help will be welcome. And
you can speak in Brazil, but Brazilian I could understand a little bit, but the other will not understand.
So if you want, Heitor, you can answer it in Portuguese and we can translate it into English, maybe. What do you think? Just to keep the discussion going.
Repeat, please.
I was wondering the effect of macrophytes on your water quality data, because there is differences between reservoir on the presence absence of macrophytes. Could you translate, Alessandra?
Heitor, a pergunta é sobre o reservatório 2, né, no qual você tem
a presença de macrófitas.
Isso, e como isso, então se poderia explicar um pouco o fato que aí você encontrou menos eutrofização do que nos outros reservatórios, junto com a presença das macrófitas.
Então, eu não estudei especificadamente as macrófitas no trabalho, mas o que diferencia a condição desses ambientes é que o reservatório 3 ele tem um maior nível de eutrofia devido a ter uma água canalizada de esgoto in natura ser descartada lá dentro, e também por receber carga pluvial do bairro todo, só que essa água não chega no reservatório 2. Então, a carga de poluição dentro desse reservatório é menor. Então, eu acho e junto das macrófitas, que são filtradoras e elas têm esse processo de influência na qualidade, em ajudar na condição da qualidade da água, elas auxiliaram a ter uma menor condição eutrófica em relação aos demais ambientes. O primeiro lago, reservatório, ele também tem uma condição mais eutrófica do que o segundo, porque existe presença de animais ali nele. Então tem patos, então tem as fezes dos animais, o pessoal vai lá e joga comida para os animais, então tem toda essa entrada além da entrada de material particulado, além do que chega por escoamento superficial da região da área de estudo.
Okay. So I tried to translate his answer. Christina, you can complement, okay? So what Heitor said, that actually the reservoir 3 is the one receives a higher input.
We have some problem with the sound, Alessandra.
What?
The connection is not good.
It's
okay. Can you hear me better?
Yeah.
Can you hear me?
Yeah, yeah.
Hello?
Yeah, yeah, yes. We listen to you, but it's like a cut, you know.
Yeah, I will use my microphone later. So the reservoir 3 is the one who receives a higher amount of nutrients, even because of animals that live in the area. And he said it doesn't specifically study the macrophyte, but he observed that with the presence of macrophyte, because they filtrate the nutrients, so they use the nutrients, the reservoir with macrophyte was the one who had the lower eutrophication level.
Okay, thank you.
E, Heitor, o seu reservatório 2 recebe também esgotos, né? Domésticos.
Não, o reservatório não. O 2, nem o 2 nem o 1.
Então
Eles recebem escoamento superficial da região, mas o esgoto canalizado mesmo, ele está direto no reservatório 3, que o objetivo dele foi ser criado como uma bacia de retenção da água pluvial do bairro, já que esse parque está localizado na região mais baixa da bacia que a gente tem ali na região.
Heitor is explaining that one of the reservoirs received untreated sewage, but was planned to receive only rainwaters, fluvial waters. This is a common problem in some urban lakes near residents. The fluvial system of the rain system is used also to throw away sewage. Então, que eu estou falando que a gente também tem esse problema que normalmente o receptor que é feito para receber águas pluviais acaba recebendo também esgoto direto. Esse é um problema muito comum. Né?
É muito, assim, porque ali a história antes daquele lago era uma wetland, era uma área de vereda. E eles transformaram o ambiente, eles transformaram a área num parque recreacional, alegando que a área de vereda estava causando alagamento no bairro. Só que não fizeram um estudo para saber se funcionaria colocar, mudar o curso, represar o curso da água e construir lagos, sabendo que ele está na região mais baixa e que toda vez que chovesse, toda a água do bairro iria direto para a região e causaria inundação, que a gente tem até hoje com os 3 reservatórios. A região alaga normalmente igual antigamente quando tinha área de vereda. Então fizeram uma transformação à toa, como se diz.
So he explains that formerly the region was a wetland, a natural wetland, and there were sometimes inundation of the region, and then they decided to change it, the region, to diminish this inundation, but they didn't solve the problem because the inundations are happening. So this is a very common problem to change wetlands, natural wetlands, into urban areas or areas that you can manage. It's a very complicated situation, but very common.
Eu não sei se essa resposta minha já serviu para a pergunta que está no chat.
Eu não posso ver o chat daqui, eu não creio, não. Deixa eu ver se eu consigo. Alessandra, could you see the question in the chat? or Bernadette. I think
What?
Alexandra is reading.
Alessandra is like mute. She has a lot of problem with the connection.
So the question is from Suvita. Says, did you hear the question, Heather?
Yeah, now
You
we
can
are
read
hearing
it
you
if you
well.
want.
Tá cortando para mim.
It's also an option
So,
to write
I
the
think
question.
Yeah, the question is written in the chat. So
Yes.
people, if you want to read it, they can just go there and read it. But so maybe we also have to move on because we have already more than
I can
half
read
an
the,
hour.
you
and
can
just
read the question, Christina?
No, it's not possible. It's not possible for me.
Yeah, because I see it on the chat, you know.
But because Christina, she's presenting, she has, because she's presenting, she cannot read it.
Not.
So, but the question, I think he already answered. It was answered already.
Okay, thank you.
Thank you, Heather. Thank you very much.
So we are going to the next presentation. Oops. Primary production in 3 urban shallow lakes at high altitude. And the presenter is Alfonso Lugo from the Universidad Nacional Autónoma de México. Three small shallow lakes of La Cantera Oriente, Eastern Quarry, an urban ecological reserve in Mexico City at 200-2,070 meters above sea level, very high, in an ancient volcanic rock extraction site and fed by springs that flow from the rock. Chlorophytes and diatoms dominate the phytoplankton in North Lake, chlorophytes in the Central Lake, and cyanobacteria in South. Gross primary production, net primary production, and respiration were measured in the 3 lakes in 2 climatic seasons, warm, rainy, and cold, dry from 2017 to 2019. Oxygen concentration records were taken every 50 minutes in periods of 8 to 14 days. And chlorophyll concentration, light intensity, and nutrients were also measured. The highest GDP gross primary production were found in South Lake and Central Lake that were characterized as hypertrophic, while the North Lake, at level a bit less, was eutrophic, hypertrophic. The net primary production attained high values in the south and in the central lakes than in the north. The range of respiration values was in the south about this range, and the central about this range, and in the north a little less, 10 to 2084. Significant differences were found between the dry and rainy seasons, and water residence time would be the most important factor causing these differences. Please, Alfonso, do you have any complementation?
Yes, thank you, Christina. Well, this case is very similar to the previous work. But in this case, we have three. Well, there are really four lakes that only one of them are completely artificial. The other three were accidentally formed because when the excavation in the quarry reached the underground water level, well, the water began to flow and then form these lakes. Later, they were reconstructed. Well, they were separated by the man, and then they are well, they are interconnected, but they are strongly managed because usually when the rainy season is heavy, the area is overflowed, and then they need to maintain the water levels. Well, we work for a long time in this area, and well, our interest in this work was to know how productive can be these lakes. They are well strong. The productivity is high, but it's comparable to other lakes in Mexico City that are filled with treated waters, with wastewater, with flows from system treatment or treatment systems. And in this case, well, the water that filled the lakes is really clear. It had well, medium levels of nitrates and well, around 150 micrograms of phosphorus. But the differences in primary production in the lakes and the trophic state are strong. No, and, well, we use this area as a very interesting site to study urban lakes because they are different from the other lakes that we have in Mexico City.
So, do we have questions?
Yes. I would like, you use two kind of methods for measuring primary production. You use
Yes.
the light and dark bottle system, you know, with the Winkler method, I think so. And you use also the sensor for dissolved oxygen. So
Yes.
There was a difference, or there some concordance or difference between the two methods?
Well, yes, we precisely we tried to know if they were comparable, and yes, the measures were comparable, and well, we think that the use of continuous measure could be useful in this case, at least, that are small lakes, very small lakes, to measure primary production. No? At least the range is comparable. We found some, well, we have a lot of data with the sensor, but many of them were very similar to those, to the less number of data obtained by the dark and clear bottles. No? But in this case, both the methods work well.
I have a question.
Yes.
Can you hear me?
Yeah.
Also?
More or less, not too much. It's
Cutting.
not now. It's not.
Perhaps you can turn off your
Video.
Your video. Maybe you can turn off your videos.
So you commented about.
Oh, so sorry, I can't understood.
Yeah, in this case, maybe it's better to ask the person to write in the chat.
Yes, please.
Hello?
Good question.
Whoever's asking the question, is it possible to please write it in the chat window?
I think you cannot hear me.
Now we can hear you.
Yes.
Can you hear me?
Yeah, very
Yes.
nicely. Yeah.
Okay. Okay. So you commented about the importance of water residence time.
Yes.
I wanted to know if you measure it, and this can help to explain the difference between the north and south.
Yes. Well,
Sorry, if you cannot
yes,
understand.
I only understand a part of the question. I will try to answer you. Yes. Well, the size of the lakes are very different, and the North Lake, that is the eutrophic or the less eutrophic, is the smallest. And also well, the flow to the different lakes is different, and the North Lake is also beside the small size receives the main flow of the channels of the springs, and then well, the stay of the water into this lake is very short, about 2 or 3 days. And in the other lakes, well, the stay is about 5 to 9 days. Besides this, the North Lake also has some more on a bigger area with microphytes that also collaborate to consume nutrients. And then this is the reason that we think that the residence time is an important factor for the difference.
Thank you very much.
Okay, I have a question also.
Yes, I'm reading the question in the chat. May I?
That's another. I'm not writing. So I wait for
well, the question is if there are a national plan for these lakes. Well, we have one very big advantage that is this area is part of an urban ecological reserve that is managed by the university. You know, the ecological reserve name is La Reserva Ecológica del Pedregal de San Ángel, and it tried to conserve very particular vegetation type that is palo loco. That is a type of vegetation that grows after the eruption of the Shirl volcano about 1,000 years ago. And then, well, in the ubication or the location of the area, de la Cantera Oriente, is into the urban reserve. And well, we are trying to propose several methods to try to manage the lakes and to obtain better trophic conditions. No, we are now just trying to use different kinds of macrophytes to obtain better trophic conditions in these lakes.
Is there any other question in the chat? May I ask my question?
Yes, of course.
You found primary production inversely correlated with water transparency.
Yes.
It was expected.
Well, yes, because well, we think that this is related to the type of algae, because, for example, in the South Lake, that is the more productive, many, well, for a long time, you can find cyanobacteria as microcystis, and then the flux well, produces a less transparency. In the other lakes, you can see different types of algae, and the transparency is better. And then more on a major portion of the water column is able to participate in primary production. This is our vision, no?
Okay, thank you.
Christina, is Vanessa. People in the chat, if you can start the sharing presentation when the people respond the question, if it's possible.
I cannot listen to you.
After to put the summary of the work, to stop the sharing this presentation.
Okay. Okay.
Or to see everyone in the room.
Okay.
If it's possible.
It's possible.
Okay, thank you.
Thank you for the suggestion. So do we have more questions? Can we go? Thank you, Alfonso, for your attention and presentation.
Thank you.
So now, retrieving chlorophyll A in a shallow productive tropical estuarine lagoon system using the Sentinel MSI imagery from Mateus Henrique Tavares is here on board. Mateus, from the Federal University of Rio Grande do Sul. Yes, I could.
Yes, hi.
Welcome, Mateus. Well, our summary on your research is your research in a estuary system, the Mundaú-Manguaba estuarine lagoon system, two connected turbid lagoons in tropical urban areas, northern Brazil. Alagoas, I think so, yeah? Yes.
Yes.
Monitoring chlorophyll A performed with remote sensing tools to evaluate the trophic state of water bodies. We validated chlorophyll algorithms using Sentinel-2 MSI, 3 dates with pairing between field collected chlorophyll data and satellite imagery between 2018 and 2019. Totaling 49 pairs of data. 7 algorithms were selected. Some algorithms performed well in Manguaba, but not in Mundaú. Combining both data sets and considering MNALS as a whole, 4 algorithms showed similar performance with a squared error of 0.77 to 0.81. And percentage of RMSE of 34.5 to 38.7%. Chlorophyll can be modeled in MNALS using a single algorithm. A semi-analytical NRE-RED ratio algorithms was recommended. Let's stop share. It's better. Well, question, please.
I can begin.
Can I just compliment?
Okay, okay.
Oh, yes, sorry. Complementation.
No, that's okay. So, hi, everyone. I think that in contrast with most study areas in this session, the system which we chose is a very large system in comparison. So it has approximately 80 square kilometers. So it's hard to get a full summary of the water quality conditions for the whole system using in situ monitoring only. So our approach was to use remote sensing tools to complement this monitoring. So we started with chlorophyll A, and with this validation, we have now starting to set a tool for monitoring this variable. with a relative good accuracy, spatially distributed, and we can generate these maps of chlorophyll A every 5 days using this information. So it's a first step in this monitoring to help management to planning and yeah, that's it.
Thank you, Mateus.
I have a question. I was wondering, you do the satellite monitoring every 5 days, yeah?
Yes.
And I was wondering if you find a nice pattern of change with the season, the dry season, the rainy season, and according to the runoff of residential area around the lakes.
We thank you for a question. We just finished validating this approach, and we only analyzed the images we used in this validation. So we observed that for the Pinel-Alloul Lagoon, which is the most impacted by the urban area, it doesn't vary really that much with the dry and wet conditions, but mostly with the tide and the nutrients that come from the urban area. So the raw sewage and storm water that runs to the lagoon affects the chlorophyll concentration. In the case of the Manguaba Lagoon, which is mostly affected by the nutrient runoff from the rivers, from the watershed, then it's very related. So it's mainly due to the cropping patterns in the watershed. So the watershed is mostly composed of sugarcane crops, and during the reaping season, you have a lot of nutrients and organic matter running to the lagoon. Even though it's a dry season, you have large, very high nutrient concentrations. It can even lead to harmful algal blooms and anopsia and fish kill events. So it's not really related to the runoff itself, but rather the conditions in the watershed and these nutrient input.
Thank you.
I have a question. Can I?
I think there are a few people with the hands raised, so I think Guillermo is the first one.
Okay.
Okay, hi. Really nice presentation, Matthew. Thanks. I'm Guillermo from Uruguay. I want to ask your opinion about if is it possible to extrapolate this strategy to other lakes as an exploration tool, and how hard, would be to automatize this as an early warning system?
Mm-hmm.
Thanks.
Nowadays, there is no global approach to monitoring water quality using satellite, mainly due to the variations in the optically active constituents in the water. So there is many different research groups working to develop this global approach in retrieving these water quality parameters using different satellites. And what we have right now is that the approach you have to use depends on the optical water types. So there is a paper that divided the water types in 13 different optical water types. And then there are another paper, subsequent paper, that evaluated the algorithms for each water type. So right now, what you have is the best approach according to each water type. So it demands some knowledge of the behavior of water and its constituents in terms of optical behavior, but it demands knowledge, so it's not easily applicable right now. And in terms of automatizing it, it's also not so easy because before you apply the algorithms, you have to correct the image for atmospheric effects. So while you can automatize the download and application of the algorithms, the automatization of the atmospheric correction tools is not easily done. So right now it's not easily applicable, especially for research groups that are not involved specifically in remote sensing. So what we are doing, most remote sensing scientists, is trying to validate this approach to make it easily accessible for the other research communities.
Thanks a lot. Congratulations for the work.
Vanessa?
I think that
it's Zali. I think it's Zali first.
Yes.
Oh,
and then
yes.
Alessandra and then Vanessa.
Sorry.
It's difficult with this virtual world.
Yeah.
First of all, I'd like to congratulate you. I think it's a really nice talk, and I think you have a great handle on the complexities. And I also want to congratulate you for your courage on working with estuaries, because they're the most crazy environment I can imagine. So my question kind of relates to the previous one. I'm just thinking about, usually estuaries are really turbid, so there's very low visibility, and with the imagery, the satellite imagery, there's always the issue of what you're measuring is just at the surface. So do you think it's necessary to calibrate what you're predicting with the satellite image from actual samples of chlorophyll just to get an idea of if you're in the same ballpark or not?
Yeah, right now we strongly recommend to calibrate the algorithms and at least validate the current algorithms there are in the literature. As estuaries are turbid, what we generally use are the bands, the spectra of the near infrared and the red. So they're not in the visible spectra, but they are less affected by other water constituents that cause this turbidity, such as chromophoric and dissolved organic matter and other non-algal particles. But yeah, unfortunately, you need a validation to at least be sure that you're estimating chlorophyll with some accuracy. You don't have to calibrate an algorithm like we did for the system we are doing, but this calibration certainly will improve your accuracy. So it's always better to have your data and to do the calibration yourself.
But I guess what I'm wondering is, do you think it's necessary, when I meant calibration, like actually going there the same day where there's a satellite passing over and you take samples at different depths and integrating that to see if it matches with the prediction of the image?
In these spectra that we use for chlorophyll, such as the red and the near infrared, the measurement is not only of the surface. It generally is a bit deeper, so you don't have to measure the chlorophyll concentration and the water reflectancy at different, forgot the word in English.
Depths.
Yes, different depths. Thank you.
Okay.
We only have a single water reflectance measurement at the surface, but still it is good to have these measurements to validate your approach.
Okay, great. Thank you. Congrats again.
Thanks. Okay, Alessandra.
So I hope it works. Every time I make a question, my system fails. So thank you for your work. Very nice presentation. My question is more or less related to what Ali asked now, but actually how deep are these lagoons that you are measuring? And because you commented in the presentation about possible bottom interference, so how this bottom interference can be corrected when we are working with very shallow lakes? So, and especially when there are lakes that are kind of temporary, they change their depth and sometimes they are really shallow. So is this something we really have to pay attention, or how important is this bottom interference?
Yes. When you have bottom interference, it completely changes your results. So you have to correct for them before you estimate the water quality parameters. So to correct for this, you need a lot of information. It's a very complex process. You need information from the reflectancy of the bottom, so the bottom sediment. So you have to do laboratory analysis to validate the optical response of this material.
So in your case, how deep are the lagoon in your case?
They are not very deep. I think the maximum depth is about 5 or 6 meters. But since they are turbid, the water reflectance does not reach the bottom, so there is no bottom effect. But in the case of clear waters, clear lakes, when this affects the water reflectance, you have to correct for this, and it is a very complex process. So,
okay, thank you. That's important. Thank you so much.
Okay, Vanessa.
Hi, Matthew. First of all, thank you for your work. It's very nice work. The remote sensing is a very powerful tool. The validation and the calibration is very important in this method. But I would like to do a question about the management actions. What are the limitations do you see using the remote sensing to monitor the eutrophication, monitoring the chlorophyll for management actions in a practical world?
Mm-hmm. Okay, thank you for your question. I think that using remote sensing is a very new approach outside the remote sensing community. So I see that this is becoming it's becoming more used in Europe, but here in Brazil, it's very, very new, very fresh. So since it's still very difficult to calibrate and validate these approaches, it is very troublesome for starting to setting up these approaches, especially when you depend on the government or public organizations to demand for these tools. So I don't remember any place where this is really applicable right now in Brazil, especially because you have this long process of validation and then you have to automatize this process to have these maps every, I don't know, 5 days, like the case of Pinel-Alloul. But we have a few initiatives in this sense. There is the HydroSat, which is an initiative by the National Agency of Waters, the ANA, which has information on a few water quality parameters for a few reservoirs and lakes in Brazil. But I think that's it. I don't know of any place that has been monitored constantly for management plans or for planning. Still only for academic purposes in Brazil.
Okay, I think the calibration and validation is a very difficult process for the agencies, no?
Yeah, it's very specific, and we have so many troubles in Brazil that this is like one of the last of the concerns they have.
Okay, thank you, Mateus.
So Mateus, I'm not familiar with this methodology, but I saw in your video the spectral curves of waters characterizing several lakes according to spectral curves.
Mm-hmm.
And I think that this methodology, we are in the beginning of the avenue. We are going to have many things ahead. Mm-hmm. And I have a question. We have this spectral curves can be as an signature of a system. So do you think that through an spectral curve, we can gain the knowledge of the features of a lake in terms of turbidity on other variables, especially the physical variation, such as suspended particulate matter and other things? Is it possible, or it's very complicated to calibrate all these results?
Yes. As I said to Guillermo, there has been a new approach in dividing the lakes in optical water types. So when you identify the spectral curve of your lake, you can try to match it to a group and have at least some general information of the characteristics of your lake. But the optical response is only related to the constituents that are affecting the optical. I'm getting some. Okay, that are affecting the spectral curves, the water reflectance. So you can have information on chromophoric dissolved organic matter, total suspended solids, and chlorophyll. So basically, there are these, and phycocyanin as well. So basically, there are these water quality parameters that you have information on. However, this curve can vary, and it will vary according to the concentration of these constituents in the water. So even for the same lake, you can have different, it can be classified as a different water type according to the season, for example. So it can be used to get this general information, but it has to be used carefully. We're still developing in the remote sensing community algorithms to try to estimate these constituents using only this spectral information. But as you say, it's still the beginning of this avenue, and we'll see. It's promising, but we're still in the beginning. I think you're.
Thank you, Mateus, for your presentation and very interesting research. I think that's the beginning of a new area new age, perhaps.
Yes.
So we are going now for the next presentation. Let me share the screen. Now will be the seasonal variation of active and inactive rotifers, Rotifera monogononta, and hatching patterns in an urban and shallow lake in Mexico City by Jair Garcia Zamorano. Is Jair here? He's not present.
No, it's not present.
Okay, let's go to the other. If he return to the session, we go back. Oops. And now, dynamics of potentially toxic cyanobacteria species and their toxins in an eutrophic urban lake, treated with lanthanum modified clay, and from Universidade Federal de Minas Gerais, Federal University of Minas Gerais. And the presenter is Bruna Barsanti. Is Bruna here? And also Alessandra.
So Bruna was here and she left. She said she had a problem with her connection, but I can't take her questions.
Okay.
So she did the presentation, but I know her work, so we are co-authors. I can take her question if she's not able to go back to the connection. I'm here anyway.
Okay, thank you. Well, the research was in Pampulha Lake, the city of Belo Horizonte, southeastern Brazil, with persistent cyanobacteria blooms, several potential toxic species. Between 2016, municipality of Belo Horizonte started a treatment with lanthanum modified clay, phoslock, to reduce phosphorus concentration and eutrophication. The study followed up the seasonal dynamics of cyanobacteria species and the cyanotoxins during 2 years field study, and investigate the relationship between the potential toxic species with the toxins detected. Signs of water quality improvement and phytoplankton structure recover in some periods, but cyanobacteria persisted. Cyanobacteria communities was mainly composed by the potential toxic species Raphidiopsis, formerly Synthropermopsis hasborski, Microcystis aeruginosa, Planktothrix agari, and Planktothrix isotrix. Microcystis aeruginosa was the main predictor of the microcystins present in Pampulha Lake. And Raphidiopsis hasborski and Planktothrix were predictors of the saxitoxins occurrence, several of them. One is that these toxins may be more widely distributed in urban lakes than has been reported, and species less reported in the literature. Alessandra, would you like to complement?
Yes, just a short complementation. That is the second part of Bruna's master thesis. The first part, she followed the treatment itself, that is this phoslock treatment to lock the phosphorus in the sediment. So what happened with the biomass of cyanobacteria and phosphorus in the lake? And she published this first part. And that's the second part that she's trying to understand what about the toxins, since the community of cyanobacteria is so diverse in these reservoirs. So there are many different species. She was trying to follow the toxins. So if a certain period of time we have one kind of toxins, and that these toxins, the dominance of one toxin over the other changes, because these toxins are very different. So the treatment, the water treatment has to be different, and also the level of toxicity can be very different, how they affect the population, the human population, but also the fauna, so fish, etc. Some accumulate in the fish, for example, much more than others. So saxitoxin are known for their accumulation more than microcysteines. And so a series of problems follow if the toxin actually changed during the year. So that mainly the basic question of her work. So now, if you have any question.
Okay, I have a question. I was looking at the result, and I found that there is a streaky difference in TP, total phosphorus, and SRP, soluble reactive phosphorus, you know, because TP decrease after the treatment, but not SRP. Could you explain it to me?
Yeah, so the total phosphorus measure the entire amount of phosphorus, so what is dissolved and also what is in the cells. So in the case of the biomass, the algal biomass in the cyanobacteria biomass. And what happens that the first consequence of the treatment is really to limit the phosphorus for the cyanobacteria. The cyanobacteria die. So the total phosphorus, the cells that go to the sediment, they die. And the first thing we observed is actually that soluble reactive phosphorus, or the solvent phosphorus, at the bottom of the lake increases. So it's like the cells die and there is a release of phosphorus, right? And so we really observed a decrease in total phosphorus, and then increase at the bottom of the soluble reactive phosphorus. The data she presented now are just the surface data, so of the euphotic zone, but at the bottom there was an increase in the solvent phosphorus. The first moment that was the consequence. Okay. And then we think that's exactly what is expected from the treatment. So they affect the cyanobacterial cells, they die, and there is the first step is a release on the solvent phosphorus. The dynamic of the two fraction is really different. We can see that.
I have another question, but I will wait for the others to give their question too. Yeah.
Well, I have a question. Well, Pampulha Lakes is a soup of toxins. It's incredible.
Yes.
Small lake, it's like a soup of toxins. So according to the results, so you studied the previous situation. So the use of the treatment was not very effective along the years. Can we conclude this?
So that was very interesting, actually, because the municipality started this program of treatment, right, with this Foslock. Foslock is well known. There are several publications with this treatment. And what we actually saw that immediately at the beginning of the treatment was very effective. So the cyanobacteria just fell, they disappeared. We even had an appearance of a huge population of Daphnia that wasn't in the lake for many, many years. It was very red. It kind of came from the bottom, so from the anoxic sediments probably. But so at the beginning was very effective. The problem, it is very serious, I think, in all urban systems. So what the municipality is trying to do, and they're trying different ways, they're trying to treat the lake, Pampulha Lake, because it's a very beautiful lake, and many people people go on weekend as a leisure, for leisure, but the problem that the input of phosphorus in the lake is not stopping. So if you don't stop the input of nutrients, any treatment you do in the lake itself is limiting, right? So what happens, the first year it works very nicely. So there were...
Oh, it's blocked.
Freeze.
Alessandra, less rain, I think. So the input of, there were several problems with...
Is the connection not working anymore, Alessandra?
Connect, yeah.
Yeah, my connection is horrible. I'm so sorry. Can you hear me?
Yeah, now yes.
Okay. So what happens that in the second year, there were problems in the sewage system. So the input of phosphorus was very high and the treatment stopped working. Can you follow?
Yeah.
Yeah.
And so that's the problem, I think, with urban lakes, that they are not natural lakes. So the anthropogenic impact is very high. And if you try to management measures, they are so dependent from what is happening in the entire watershed. And Pampulha watershed is more than 50 times the size of the lake. So external impacts are huge. And so the treatment itself is very limited because it depends on many, many other things.
Okay. Thank you, Alessandra.
I see there are some questions. Should I answer them?
Yeah. Yes.
So there is a question here from Christian Alberto. Said, interesting work. Do you find an environmental variable associated with cyanotoxin? And then do you think there is a negative effect of false lock on Benton community? So the first question with the cyanotoxin increase, actually, what we are looking now is just the connection between the cyanotoxin and the cyanobacteria species. So if there is a control of the cyanobacteria species present, we will control also the cyanotoxin. But your question is very interesting because I will have a student to try to see if the false lock somehow can select for species. They are more adapted to the new condition and somehow they may produce more or less toxins, you know? That is a lot about the selection. So false lock actually is selecting some genotypes over other genotypes. So genotypes, they are more adapted to the new condition. They could be more toxic or less toxic. So that is something we want to investigate. It's a very good question. About the benthic community and the false lock. False lock is known in the literature as being a neutral chemical. So it's just like of a silt. So it's like something that's neutral for biological community, has been tested a lot before having permission to be used in lakes. And so theoretically, from what is published, it shouldn't have any effect on the biological community, you know, except the fact that it's removing phosphorus from the water. And so there is less phosphorus for all the photosynthetic organisms to grow. So thank you, Christina. The other question is from Fernanda, and she asked, congratulations on the work. Cyanobacteria are no longer dominant in the community. How long afterward did the concentration of toxins start to increase? So actually what happens is the cyanobacteria at the beginning of the treatment, it was really amazing how the cyanobacteria, they disappeared. But now they came back, right? So somehow the treatment is not enough really to stop the growth of cyanobacteria. And as Christina said, this lake is really a soup of cyanobacteria. There is a huge biomass of cyanobacteria. And so the treatment, as it is now, after the initial success, it was very successful. Now the cyanobacteria are back. So not so high level as before, but they are back, unfortunately.
Okay, thank you.
Okay, she has another question here. How long afterward did the concentration of toxins start to increase? Actually, the concentration is still high. So we didn't see a decrease and an increase. They are still very high. Okay. Macrophyte. Yeah, that's a good question too. So I will answer just the last question further. We had, I think, the same as Christina will show in her presentation later. And so Bernadette also said, the macrophyte, there was a time when the macrophyte, they covered large part of the lake. During this time, we didn't have blooms, and the amount of phosphorus was much lower in the lake, but the macrophytes were removed because we had huge problem with mosquitoes for the population, so they were entirely removed. And there should be a very nice strategy, a very good strategy, but should be controlled, of course, because can also be a problem, but would be an interesting strategy. Thank you for the suggestion. Yeah. So thank you.
Okay, let's go for the next. Linking internal phosphorus loading and macrophytes distribution in a shallow reservoir under restoration, Western Poland. And Renata Dondajewska-Pielka is here. Or other collaborators.
Yes, I am here. Hello.
Oh, thank you. Hello. Welcome, Renata. So I'm going to present a brief summary of your research, and you can complement if you want, if you wish. Rusałka Lake, a man-made reservoir in the city of Poznan, Western Poland. Water quality deterioration and cyanobacteria blooms. Restoration measurement treatment, small amounts of iron sulfate added several times per year from 2004, 2007, 2014 and forward. And chloride of magnesium 2040 and forward. Submerged macrophytes harvest 1 year since 2017. Macrophytes decay in late summer followed by appearance of cyanobacteria blooms. Sediment cores, triputate, were collected in profound and littoral zones. Sediments exposed to controlled conditions, temperature and oxygen. Observed decrease in internal P-loading in both regions due to increased sorption complex in the sediment. Macrophytes, changes of internal loading due to stabilization of sediments and P uptake, decrease in loading, and the composition of macrophytes on the sediments, increase loading. So Renata, would you like to complement something?
Yeah, thank you for that comprehensive summary. Maybe I will add one more information. In fact, we have a problem with quite high internal phosphorus loading before the restoration when there were no macrophytes in the ecosystem. Now this problem of internal loading is still present only in the part of the year, the second part of the year in summer, when after the macrophytes decay. So we observe that the positive effects of presence of microphytes on one side, but on the other, when the microphytes decay, still internal phosphorus loading is observed. Therefore, the future management should go in this direction to increase the period of macrophyte presence up to autumn. Probably with different species, not only Potamogeton crispus, because it has a quite short time of presence in the ecosystem. But yeah, in future probably we will work on increasing the amount of species in this reservoir.
Questions? Guillermo?
Thanks, Christina. Renata, nice to meet you. That's a really interesting presentation.
Hello.
Thanks. Hello. So, two questions. We are searching for options to work with eutrophic lakes here in subtropics. And iron compounds are very sensitive to redox conditions of water column and sediments. So we are not thinking in iron. And my question is about how is the thermic dynamics of the lake that you are working on, and how much hypoxic or anoxic are the lake? Is the lake? Thanks.
Oh, okay. This reservoir, in fact, most part of it is shallow, about 2 meters of depth. So there is no problem with toxic conditions. The anoxic part only is only the deepest part. This only few hectares, few percent of the whole reservoir. Therefore, we decided to use this iron sulfide in the beginning because we knew that only in the deepest part, this very small part, in that part, the iron sulfide might not affect as we expect, but in the other, on the most of the reservoir, it should work very good.
Okay, thanks. Another question. What about magnesium chloride? What was your experience with this compound?
Yeah, we're just starting working with it because it's quite cheap. In fact, we deal with problem of funds in case of restoration in Poland. Therefore, we are searching for as much cost-effective methods as is possible. I must admit, I am amazed when I see reservoirs where lots of phosphate, for example, can be used. It's definitely too expensive for Polish lakes. In case of magnesium chloride, the idea is to precipitate not only phosphates, but also ammonium nitrogen. And we are using this compound not only in Rusałka, but only in other lakes, and we can observe a positive results in both in case of ammonium and in phosphates. The content is lower.
Thanks a lot.
No problem.
I have a question.
Yes?
You found that the phosphorus loading was lower in the littoral zone in 2007, and comparing before and after treatment, you know?
Yeah.
And is it due to the presence of macrophyte in the littoral zone?
Yeah, after 2014, yes, definitely yes, because there were no macrophytes until 2007, and this moment we still observed internal loading, but after macrophytes appear in part of the year, we could observe that the internal loading was lower or even negative, which means that the phosphorus getting into the sediment, not from the sediment to the water. But definitely in the shallower part, most part of the reservoir, this was the result of the macrophytes.
No. Okay, thanks.
Thank you.
I have a question.
Okay.
So from your presentation, you found a correlation between phosphorus release from the sediment and temperature, also related to organic matter. And so you have higher release of phosphorus during summer, right? And then when you have the higher temperature. And in summer is exactly also when you say that the blooms start to appear. Do you think that there is a connection with this increase of phosphorus, so increase of temperature, phosphorus release, and cyanobacterium bloom?
Yeah, definitely. Because when summer is in the beginning of summer, we usually observe that there's a very serious, strong decay of the microphytes, and therefore the nutrients are going into water. And for sure, the akinets of the cyanobacteria are in the sediments. So it's a perfect moment for them to start coming to water and create blooms.
Okay, thank you very much.
Thank you.
Do we have more questions?
Yes, I have a question.
Please.
Well, I would like to know if you observe changes in the pH or in the community structure of zooplankton after to add those substances.
No, I don't recall any of that in case of pH. In case of zooplankton, yeah, I must admit that we are doing the analysis of zooplankton, but we haven't done any statistics or any analysis of that yet. It will be done for sure, but at this moment it's hard for me to say is there any tendency. Before the treatment, before the microphyte covered in the zooplankton, there were mainly rotifers. But I assume that in the part of the year when microphytes are present, probably the amount of, for example, Cladocera should increase. But our results definitely need some analysis in this area.
Thank you.
Thank you.
Okay, let's finish. Thank you, Renata.
Thank you very much.
We have to increase our speed according our delay. Next presentation. Dominance of phytoplankton or floating aquatic plants in a hypertrophic urban lake, consequence for carbon and nitrogen content in the water and for plankton communities, is myself, Christina Castelo Branco, Karen, a student of undergraduate, and Samira Portugal, another professor of Unirio. Well, Lagoa das Taxas is an hypertrophic shallow lake inside a natural park surrounded by residential buildings in Rio de Janeiro that receives untreated urban drainage and is a refuge for wildlife. Mechanical removal of floating plants once a year. Two alternative conditions: complete coverage by macrophyte or phytoplankton dominance. Sampling dry and rainy season from 2014 and 2019, when aquatic macrophytes dominate, anoxia, drop in chlorophyll A, chlorophycians and cynophycians dominate the phytoplankton. Without macrophytes, chlorophyll A attained higher values and granophycians and or filamentous cyanobacteria prevailed. Zooplankton, small-sized testaceans, ciliates, rotifers, cladostrans, and cyclopod copepods, species highest species, richness higher under phytoplankton dominance. Mechanical removal of plants, improved the dissolved oxygen, decreased concentration of total carbon and total nitrogen, and apparently no effect on total phosphate content. Mixotrophic taxa in phytoplankton under dominance of aquatic macrophytes, intense reproduction of rotifers in favorable conditions, accessory pigments in microcrustaceans during anoxia, common presence of reproduction in Cladoceras with the production of Ephipia. Sorry for the speed. We are in a little bit hurry. So questions?
I was wondering, the brownish color of the zooplankton, did you test if it is like a pseudo-hemoglobin protein, something like that? Coloring the species?
We didn't test, but according to literature, the moina, the cladocerans, the moina especially, they have the capacity to produce hemoglobin in the anoxic conditions. To our surprise, we found brownish color also in rotifers.
I'm in a conference meeting.
So
I will come at 11 later.
It's a new finding, and we must, I think, do some analysis searching for hemoglobin in rotifers. I don't know.
I have a question, Christina. You showed actually a higher richness of everything, phytoplankton, zooplankton, under the phytoplankton situation. And often we found the opposite actually, because macrophyte, they kind of increase the number of niches and habitats and things like that. I was wondering, because you showed those pictures that the macrophyte, they really cover the entire water surface. It could be related to total absence of light for the phytoplankton and then for the zooplankton. Could this be a reason that you have lower richness and the mixotrophy at the same time?
Really, when we have the condition of dominance of macrophytes, we have low values of oxygen in the water, as well very rich communities of predators, insects, and libellulidae, nympha and larvae, and others, dipterans. They fed upon zooplankton. So with macrophytes, increase the diversity of the aquatic system, but for the zooplankton, it's not very good condition. I think that especially because of the anoxic condition. Okay.
Thank you.
Do we have more questions?
That you answer already to the question about I was wondering, you have a lower diversity of zooplankton with macrophyte, which is totally inverse that we found in the temperate lakes. When you have macrophyte, you have higher biodiversity of zooplankton. But you gave me the explanation before, you know, because of the anoxia and because also of the insect predation, you know.
Yeah, it's a very...
Yeah, it's mainly due to the difference in the kind of macrophytes. You have Jacinto, you know, Eschnoria, that is covering all the surface. Here in the temperate lake, we have submerged macrophyte, but not as the same as you. Yeah.
Yeah. Yes, we have production all the time, all the year. It's the...
Yeah, yes, the difference.
Yeah.
There is a question in the chat, Christina.
Oh, guys. Melanism in zooplankton has been related to high UV radiation. Do you find any other reason to this pigmentation? From Christian Alberto. Well, I think that in the kind of melanisms related especially to anoxic condition, it's different. It's not a protection for UV. It's an increase in the capacity of respiration to increase the content of pigments capable to transport oxygen. So I think that's different targets to have pigmentation. And do you consider zooplankton sessile species attached to macrophytes? I think species richness would increase on macrophyte beds. Yes, we took sample of the phytoplankton, but in these beds there are many mollusks, and they are fed on this periphyton. So we only have small bedyloidans in this macrophytes beds. So the richness is very, very, very low, really. I don't think that I answer your question, but is this? Okay, thank you. So can we go to the last presentation of our session? Oh, share screen. So, rehabilitation of eutrophic subtropical urban sandpit lakes in Canelones, Uruguay. First 10 years following an adaptive management approach from Guilherme Goienda from Centro Universitário Regional del Este, Universidade de la República. Guilherme is here, I saw him. So, artificial lakes in Ciudad de la Costa, Canelones, Uruguay, remains on sand extraction started 70 years ago. Strong anthropogenic pressures, including fish kill in 1986. And in the 90s, the area began to change from vacation to residential, including an explosive population growth. Environmental management started in 2008 with a water quality strategy plan defined by the municipality in collaboration with the Université de la República. As a result, the diagnostic eutrophic state was generalized and detailed information about the ecosystem stated and functioning was generated. The management interventions included macrophytes harvest, nutrients immobilization, and destratification. A permanent monitoring program was implemented, including interinstitutional partnership and integrating high school students and teachers and trained neighbors. 3 telemetric monitoring buoys were installed, assuring open access to the data, and next goals include numeric modeling of ecosystem process and responses. So Guillermo would like to complement something.
Thanks, Christina. Yes, I have to say that we miss one co-author of this work, that is Martin Pacheco. We have 2 people with the same Pacheco surname, and some kind of problem. We miss one of them, and please, sorry. As you can see, we are a big number of limnologists, mainly, that we are working in these lakes with the support of the municipality and strongly in relationship with the community, and try to put the knowledge and the research to improve the environmental condition of these systems. So I'm open to the question if there are any. Thanks.
Yeah, I was very impressed by the high technology you applied for the management and the monitoring. And I was wondering, did you use all the data you get to try to do like a model of environmental control in this lake, based on the physical parameters that were the most variable and the most discriminant, you know.
Okay, we started to the automatized monitoring last year, and now we have two students, master students, that will work with this information, and we make a work. Sorry for my English, but we are talking with a professor from Japan that he has experience working in numeric modeling, and we are trying to understand first the thermic dynamics of these lakes that are almost all of them are monomictic, and it's not really shallow lakes, most. And after that, we are also trying to understand the effects of a reaeration oxygenation system with air pump. And the application of this information for management is we are trying to apply this knowledge in the future, but now we are starting work with.
Yes.
And I don't know if I understand completely your question, Bernadette.
No, you answer very well, you know, yeah. You are at the beginning of the monitoring, and then after you have to extract the more discriminant parameters and to do a model with them, you know.
Yes, yes. We have two different systems, technological system. One of them was designed for our group and developed in Arduino. It's a free platform. And in this boy, we have the capacity of measure temperature each half meter of that in one system. So in this case, Laura Lopez, who is the master student, will try to understand how the thermic dynamics of the lake. And the other two boys are different, has sensor in the surface and in the bottom, and include temperature, pH, oxygen, turbidity in surface, redox. I'm missing something, but this system is not developed by us. The sensors are Aqualabo is the brand, and the telemetry and all the other things were developed here with a private enterprise.
Because in our group of research, we're doing this with boys on a series of lakes to follow on the long term the effect of warming in the climate and all the technological parameters that are changing with the climate warming. Yeah. Thank you. Very interesting.
For you, thanks. We will try to maintain these stations working for a long time, and try to see these kind of things that you comment. Yes,
yeah. Thank you.
Thanks.
Any question?
I have a question, because I found very interesting that you focus at the importance of the interaction between politicians, managers, and academics. And I just wanted to know about your personal experience. How open really are the managers and politicians when the academic comes with something that is extremely important, but sometimes expensive, sometimes long-term? How open and how was actually this relationship you had with this people?
Close your microphone, Guillermo.
Thanks, Christina, and sorry. Thanks, Alessandra, for your question. Yes, I think we had a very particular conditions to reach these objectives with this project, and in this case, from both sides of this interaction between management and academy, we also were the same actors, because some kind of politics stability in this region puts the same person in the same position to take decisions. So, and in the university side, we was trying to help to some environmental problems and working with the community, and we make some kind of we have the same or complementary objectives. So in these conditions, we were able to make all of this in something like more than 10 years. But it's very difficult to think if this will continue in the time, if the conditions in one or the other side change. And this is, I think, we have to think how to solidify this experience to maintain in the time without depends on the person of the person that's using each chair. But I think the experience reach a level that is very difficult to broke now, but always is possible. Always is possible. So we will see. We are working for making this possible. We put a lot of effort and time in on this, and but we have to continue working to that. If not, this will finish tomorrow. Thanks.
Thank you. It's a very important connection, what you say. Without this connection, it's very difficult. That's another problem of urban lakes, no? Because we do have the municipality to work with, to interact. So it's a connection that we need to keep going, right?
Yeah.
Very nice. Okay, thank you.
Okay, sorry, another comment. I think the open mind have to be in the both sides of the interaction, because as a scientific, we are working in the sharp side of a knife always, because the problems are big, the community ask for solutions. We don't have a book of recipes of how to solve these problems and in a cheap form. And there are another things more far from environmental things that you have to be in consideration to give opinion to managers and community. And it's very difficult to work, but it's really interesting. It's really interesting. But a lot of headaches associated with this result line. I'm interested in alligators from Rio de Janeiro to put in our urban lakes. Yes, really interested. We need a bottom-up, a top-down control in these systems to some neighbor and other people. Please, Christina, send me.
Not very hot for our alligator. Oh, I have a question, but first of all, I would like to congratulate you for your excellent timeline that you present of the management. I think that your timeline gives some kind of hope for us that we can do, and we have to follow a way, a line, perhaps a timeline. I have a simple question. I have two questions. The first one, the beautiful pictures, did you take them by a drone?
Yes.
Oh, fantastic. And the second.
I got, sorry, Christina, I can go to Rio de Janeiro and take some picture for you if you want.
Please.
Please send me the air ticket and I can go.
Pandemic, yes. Another question. Your system that came from sand extraction is very common, very common.
Mm-hmm.
And when we put together with urbanization, we have problems. And my question is about the sewage. Is there input of sewage directed into these lakes?
No, directly now, but this city grows in sand with septic tanks that filter to the water table. So all the nutrients were distributed by the water underwater, and the eutrophic problems are distributed and not only related with the lakes near to the houses. It's a general problem to all the underwater, and with the time, with the years, the accumulation of phosphorus in the sediments was bigger and bigger, the internal load. So the symptoms and the problems of eutrophication is increasing with time.
Okay.
Sorry, and some years ago, the municipality and other national companies started a big and millionaire septic system with for all the city that treat and send the sewage to the Rio de la Plata with I don't know in English, but it's a pipe, a long
pipeline,
100-meter pipe that send all to the Rio de la Plata. But in our conclusion is the underwater is so much is rich in phosphorus and will take decades to lower these levels and to take a possibility to control the input, the phosphorus to these lakes. So we can't cut the input of phosphorus to this lake. We have to work with these lakes, assuming that the phosphorus will continue arriving to these lakes.
Okay, thank you, Guillermo. I write down two words or two expressions for your work: perseverance and keep working. I think that we have to finish our session now. I would like to give the word for Alessandra and Bernadette to finish. My finished message, my ending message of this session is to everyone: perseverance and keep working in our urban lake. Alessandra and Bernadette, please. We have to finish the...
Yes, yeah. I want to thank all the participants for their nice presentation and for the various topics we have together that expands our perspective on urban lakes, you know. Thank you very much.
Thank you, Bernadette.
So thank you to all of you to have participated, to have sent your papers, your presentation to this session. The original session was organized by Christina, Bernadette, and myself, which we joined later. And I think the importance of this system are very clear. Because they are really more and more important in the urban landscape. And I think this session was very successful to show this. So the applied ecology side and the theoretical ecology side too, right? Because they are so ready to change in these anthropogenic conditions. So thank you very much for all of you, and to Christina to have organized this session.
Okay, so thank you all, and let's see in other sections. Bye-bye. Have a nice
Thanks a lot.
Bye.
Bye-bye.